Hello everyone, welcome back to the channel Microbial World. So in the previous class we discussed the structure and symmetry of virus. Today we will be studying about bacteriophage, its structure, and the viral replication cycles, lytic and lysogenic cycle. So bacteriophages are bacterial viruses that infects bacteria. Bacteriophages was first observed by Frederick W. Twort in 1915. The term bacteriophage was used by Felix D. Herrell in 1917. Phage structure. Bacteriophages are usually double-stranded DNA viruses. The head region is surrounded by a protein coat called capsid, which is made of small subunits called capsomeres that protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. The capsid of a virus with the enclosed nucleic acid forms the nucleocapsid. Neck is the region that connects the head and the tail, and it consists of a collar or disc and several whiskers. Tail is the narrow cylindrical part. It has a central hollow core or tube through which viral DNA is injected into the host. The core is surrounded by sheath proteins. Base plate is the hexagonal plate with tail fibers, usually 6 in number, and used for attachment to the host cell wall during infection. Tail fibers can recognize specific receptor sites in the host cell wall. Replication. There are two different types of viral replication or life cycles: virulent or lytic cycle and temperate or avirulent or lysogenic cycle. Lytic cycle. Lytic cycle or vegetative life cycle causes the lysis or rupture of the host cell and release of large number of new viruses. Bacterial viruses exhibiting a lytic life cycle are referred to as virulent bacteriophages because it causes the death and destruction of the host bacterium. Examples include T-even phages T2, T4, T6. The lytic cycle consists of 5 steps: attachment or adsorption, penetration or injection, synthesis or transcription, assembly, release or lysis. The first step of lytic cycle is adsorption, which involves the attachment of tip of the virus tail at the bacterial cell wall surface via specific receptor site. The specific receptor of the bacterium is lipopolysaccharide, although any surface structure functions as phage receptor, including flagella, pili, and carbohydrates. The second step is penetration or injection. Penetration is facilitated by the digestion of cell surface structure either by phage enzymes, for example lysozyme, or by viral activation of host degradative enzymes. Penetration is achieved when the tail fibers of the virion attach to the cell and hold the tail firmly against the cell wall. The tail sheath then contract and drive the tail core into the cell through the cell wall. The virus then injects its genetic material into the cytoplasm. The third step is synthesis. Once the bacteriophage genome enters the cytoplasm, the phage DNA initiate the synthesis of early proteins. Some early proteins break the bacterial DNA and take control of bacterial cell machinery. The other early proteins serve as an enzyme for the replication of phage DNA. The newly synthesized phage DNA produce late proteins, the protein used for phage capsid. Fourth step is assembly or maturation. After the synthesis, capsid protein assembles to form empty head, and viral DNA is packed inside it. This results in the formation of numerous intact phage particles within the cell. The fifth step is release or lysis. After the assembly step is completed, viral proteins cause the lysis of host cell and all the viral progeny are released into the environment. Lysogenic cycle. In lysogeny, the viral DNA of the temperate phage, instead of taking control over the function of host cell machinery, the viral DNA is incorporated into the host DNA and becomes prophage. A cell that contains prophage is called lysogen. In this situation, the bacteria metabolize and reproduce normally, the viral DNA being transmitted to each daughter cell through all successive generations. Sometimes when the lysogenic bacteria are exposed to UV light or chemical, the viral DNA is removed from the host chromosomes and the lytic cycle occurs. This process is called spontaneous induction. Example of avirulent phages are lambda phages. So during the lysogenic cycle, the phage first infects a cell. The phage DNA becomes incorporated into the host genome. This is called the prophage. The cell divides and the prophage DNA is passed on to daughter cells. Under stressful conditions, the prophage DNA is excised from the bacterial chromosome and it enters the lytic cycle. The phage DNA then replicates and phage proteins are made. New phage particles are assembled. The cell lyses, releasing newly made phages. So this is all about the viral replication cycles, that is lytic and lysogenic cycle. Thank you.